Dear colleagues, it's my pleasure to be invited to this BME Horizon lecture. New Engineering journal, which I think addresses important needs at the interfaces of medicine, biotechnological tools, engineering, imaging, etc. So I'd be very happy to have a good and engaging discussion today on what I think we can do with advanced engineering strategies to help improve the performance of nanoformulations for the diagnosis and treatment of cancer. So I work at the Institute for Experimental Molecular Imaging in Aachen in Germany. We're part of the university clinic in Aachen. We are actually located at a separate institute, which is called the Center for Biohybrid Medical Systems. And we have a very interdisciplinary team with people from pharmaceutical technology, from medicine, biology, chemistry, material science, various different backgrounds, all basically contributing their individual expertise towards multidisciplinary solutions for tough treatments, because cancer is a very difficult disease to treat. I'll start with one or two slides on really the fundamentals. I guess to most people in the audience, these things will be known. When we talk about cancer nanomedicine, we typically refer to carrier materials, to drug delivery systems. Some people define them as 1 to 100. I think by the broadest of definitions, it should be 1 to 1,000, right? Also a 200 nanometer particle should be called a nanomedicine formulation. And what they do is essentially these three things. So they, on the one side, enable the use of difficult drug molecules. Molecules like mRNA, for instance, would never be able to work as a drug molecule if they wouldn't be encapsulated within a very compact nanoparticle. RNA is very labile, very unstable. It's rapidly degraded in the body. It's also highly inflammatory if you inject it. So without a formulation, it cannot be a drug. And the same holds true for highly hydrophobic medications. For instance, some of the taxane drugs that are extremely extensively used, paclitaxel, docetaxel, cabazitaxel, all of those need to be formulated in quite toxic solvents, formulation aids essentially. But instead of using things like polycastor oils and chemophore, we might as well use things like polymeric micelles or certain types of lipid nanoparticles to entrap such hydrophobic molecules in a nanocarrier that can then help to overcome these two aspects. Right? So on the one side, we want to protect the drug from the body. We want to prevent drugs from being degraded in the bloodstream and in the liver, from being excreted very rapidly via the kidney into the urine. But at the same time, we want to protect the body from the drug. So we want to prevent that toxic drug molecules end up in organs where they cause a lot of damage, like the heart for anthracyclines, the peripheral nervous system for taxanes, the kidney in the case of platinates. All of those side effects can be bypassed if we formulate agents in things like liposomes or micelles or conjugate them to antibodies, etc. And then from a strategy point of view, like if we think about different targeting strategies, clinically, by far, the most widely used is what we refer to as passive targeting, which is based on prolonged circulation of nanoparticles connected to endothelial leakiness. It's typically referred to as the EPR effect, but now there are alternative mechanisms like the ETR effect or the ATR effect, which also consider transcytosis, which is definitely also a mechanism of nanoparticles to enter the tumors, which is through the cells, not in between the cells. And then we also have a retention component. In the old days, people believed that nanoparticles are retained in tumors because of a lack of lymphatic drainage. Nowadays, we know it's due to the presence of tumor-associated macrophages. Right? Macrophages take up nanoparticles in tumors and they keep them there. And then we can do multiple things. We'll talk a little bit about some of those. We can add targeting ligands to the surface of the particle. Many people think that adding a targeting ligand will get more particle to the tumor. That's in 9 out of 10 cases not correct. What it does help to do is to get the particle into the target cell, which is not a macrophage normally, but a cancer cell. Right? So you basically shift the balance with active targeting from macrophages to cancer cells. Then we can do triggerable formulations, either hyperthermia triggered or ultrasound trigger. I'll talk quite a bit about this in the second half of my talk. And we can also target to endothelial cells. I have 1 or 2 slides on this concept as well. If we look at the current clinical market globally, by very strict definition of what a nanomedicine is, and in this figure, I excluded antibody drug conjugates. There are 17 antibody drug conjugates approved at this point in time. Not all of them, obviously, like nanoparticles in all countries, but in certain countries. antibody drug conjugates are gaining a lot of traction now in large pharmaceutical industry. There are many, many trials ongoing because we finally managed to get them to work properly in patients. And I am 100% sure that in the next decade, we'll be seeing the same for nanoparticles, right? We already have a good number, but in a way, this doesn't really weigh up to the large number of publications, and particularly also to the very, let's say, blunt or overly optimistic claims that we have seen in some of the nanomedicine literature. So if you look at the formulations we have in the clinic, obviously many of them are liposomes or lipid nanoparticles. Some of them are inorganic particles like iron oxides or hafnium oxides, which are used to enhance physical therapies. Iron oxide nanoparticles can be used to produce hypothermia, so to thermally kill tumors. Hafnium oxide nanoparticles, they potentiate the effect of radiotherapy. And then one of the coolest things, I will not talk about this, but we have a big interest in this in the lab, is to use nanoparticles for multi-drug delivery. So one of the exquisite things that you can do with a nanoparticle is load an exact ratio of drugs in the formulation. In this clinically used leukemia formulation, it's actually a 5-to-1 ratio of cytarabine to daunorubicin. So you have something like 5,000 molecules of cytarabine, 1,000 of daunorubicin, and at that fixed ratio, they produce pharmacological synergy. That means that the effects are more than additive. So 1 + 1 gives an effect that is more than 2. And those effects are typically only maintained at strict ratios, like this 5-to-1 ratio. And if you administer those drugs to the patient intravenously or orally, to ensure that you can keep that ratio also in the tumor cell, it's essential that we co-encapsulate them or co-conjugate them to a particle, right? Or to a polymer or to an antibody. Otherwise, they would distribute differently in the body. They would be excreted differently by different organs. They would have different modes of cell uptake. They would never maintain the ratio at which we administer them to produce that synergy. So this is something we'll definitely see more of in the future. There's also been criticism on partially the number of papers produced versus the number of drugs that we have actually used in the clinic in patients. People have worried over overly optimistic developments of certain actively targeted nanomedicine, and other people ask the question, so what went wrong with the design and how can we make it right? And I'll discuss a couple of strategies with you that we work on in the lab in which we try to make things right. For the talk today, I didn't want to overwhelm you, so I'll only focus on 2 different aspects that we work on. These are, I think, very promising aspects that in general in the nanoparticle field we should be working on. We dubbed them back in the days as SMART, with SMART not being responsive to triggers or typically be like highly elegant multi-component formulations where it's actually almost an art how people can make such multi-component formulations. But from a clinical point of view, SMART typically means simple, right? Simple solutions for day-to-day problems where you take a nanoparticle, you target an immune cell, and you boost the immune response. Or where you take a particle to improve or change the biodistribution of a drug to enhance combination therapies, right? Or where we load combinations of 2 different drugs. Instead of doing 1 drug per particle, we can do smart combinations of 2 drugs. What I do think though, and I think the first part of my talk is kind of boring, but I think it's really important to understand that the key reason why nanoparticles haven't had more impact on the clinical treatment of cancer patients is because we haven't figured out how to find the right patients to treat. In antibody treatments and antibody drug conjugate treatments, what people do is they take a biopsy of the tumor, they stain for the receptor that the antibody binds to, which can, for instance, be HER2 or EGFR, and then only those patients that express that receptor will receive the drug, right? Out of 100 patients, that's probably 15. That means 85% of patients do not get the drug, but in the 15 that we do give the antibody to, the drug is quite likely to work. What we still do, right? And this is for antibodies, this has been done for more than 20, 25 years. What we still do in nanomedicine is we give the drug to all 100, hoping that all 100 will respond, and that is not realistic, right? So we need to figure out ways how to find the right patients. Before we move to these solutions, I want to make sure that everybody is aware of 2 different things. One is that numbers do not really matter, right? Numbers matter in many regards, but not per se when it comes to a percentage of a drug or drug delivery system that goes to tumors. In a very famous meta-analysis published in Nature Reviews Materials many years ago, Warren Chan and colleagues, they analyzed how many particles of 100% injected into the bloodstream eventually make it to the tumor. And they found basically for every year, they looked at more than 200 data sets, that essentially only 0.7% of what we inject makes it to the tumor. And if we would be able to raise hands now and see who thinks this is a good number, I don't think anybody would raise their hands. Right? And if it's a bad number, probably many people would raise their hands. But the point is, the number does not matter. This number is not bad. Free drugs are much, much lower than 0.7%. Antibody drugs in the clinic, we wrote a comment on this, asking, what is the point? Is the point targeting or is the point treatment? Right? We should not let ourselves be misled by numbers, because numbers do not define whether a drug works. This is a study, or this is a slide from a famous Dutch oncologist, Liesbeth de Vries, presenting at the biggest clinical oncology meeting in the United States, the American Society for Clinical Oncology, a slide on the amount of antibodies that accumulate in tumors in patients. So this is clinical data, 4 different antibodies. All of them are used in the clinic every day to efficiently treat many patients. And those numbers are all below 1%. It's 0.1, 0.4, 0.3, 0.9. And still, they all work. They produce a lot of good effects in many patients because they're given to the right patients. Because people would have stained for HER3, right? Or people would have stained for mesothelin. So they find the right patients, and then 1% is more than enough to do the job. So 0.7% does not mean drugs cannot work. I think that is a very important take-home message for everybody. The other thing is, people have said that this, what we call the EPR effect, or this tumor targeting effect, that it fails in the clinic, that it only works in mice and in rats, not in humans. It cannot be proven in the clinic. This is published in Journal of Controlled Release, which I think is a really good journal. Many, many citations, and the message is completely wrong. Right? If you look at the clinical situation, and I'll show you some own data later on. This is data from classical studies. Like there are very, very few imaging studies, quantitative imaging studies available in patients, unfortunately. But what we knew from those studies is that if we look at sarcomas, angiosarcoma, Kaposi sarcoma, fibrosarcomas, Ewing, all of these situations, they show liposome targeting to tumors. Here's a head and neck tumor for targeting. This is a lung tumor. This is a breast tumor. Right? These things here, this is the heart, cardiac pool, liver and spleen. Liver and spleen we cannot avoid. They're very big organs. They have a lot of blood and perfusion. That's why they're so dark, so red in color. And then on top of that, they also have a completely permeable vasculature. So they're basically the same as an ideal tumor. So it's almost delusional to think that we can avoid liver and spleen uptake. The only thing we can do is try to delay it to such an extent that there will also be uptake in tumors, right? So to initially prevent uptake in liver and spleen, avoiding, let's say, capture by Kupffer cells, to allow nanoparticles to reach tumors. And if we now do that in 17 different patients, this is data from Kevin Harrington and colleagues, already quite old, but I think very insightful. What they showed is in 17 patients, if you look at tumor uptake of liposomes, 15 of them are positive. That's 88% that show liposome targeting to tumors. So it's nonsense to say it doesn't work. The problem is, though, that these numbers, the overall efficiency, even though numbers per se do not matter, but there is a big range from 2% of the injected dose per kilogram to 50%, 46%, etc. So if we would want to design a clinical trial in such a way that we can be optimally efficient, probably the best thing to do is only select patients that are 20% upwards or maybe 10% upwards, and then only treat those 30 or maybe 40% and not the rest, at least initially, until you make it to the clinic. And then later on in the clinic, you can maybe open up and also take everything above 5%. But it's so difficult to get into the clinic nowadays with all the competition and all the good drugs already out there, that you have to be sure that you treat the right patients. And imaging is one of the ways how we can do this. So I'll share with you some examples. I will not go into too much detail on the formulations themselves. I'd rather focus on the concepts. We've done a lot of work with Christiane Rijken, Crystal Therapeutics in the Netherlands. 15, 20 years of collaboration on a polymeric micelle platform, where the trick is that you actually cross-link a micelle after the micelle has formed. You use a cross-linker, and you have the drug on the cross-linker via a cleavable linker. So the whole system is colloidally stabilized, right? It will not disintegrate even if you dilute it way below the CMC because it's chemically cross-linked. By tweaking polymer size, we can tweak the size of the micelles. By choosing different linkers, we can control drug release. It's a very flexible platform. It might sound complex, but it can be easily upscaled. This has been tested in more than 100 patients. It's very robustly synthesizable and manufacturable. So it's a very good platform that I think performs really well in mice and in patients. What we see in mice, we use 41 model a lot as a reference model. 41 triple negative breast cancer is used in approximately 25% of all preclinical cancer studies. It's really by far the most used study. We did a recent meta-analysis, which was published in Nature Nanotech, on multi-drug combinations. And there we actually found that I think 25% or 24.1% of preclinical studies is 41. Second best is MCF7 with, I think, 9%. So it really dominates the field, and it's an excellent reference model to compare different formulations to each other. If we take normal pegylated liposomes, like the equivalent of Doxil or Calyx, we achieve approximately 8 to 10% tumor accumulation. If we take those mice cells that are chemically cross-linked, that are 100% pegylated, and because of that do not build a protein corona, we get up to 20% tumor accumulation, right? In vivo, this is transillumination imaging. This is not an IVIS. This is a former MI Labs device in which we transilluminate and then we use computed tomography. That's why you see the bones to reconstruct the optical signal. You can really see how efficient tumor targeting is with these mice cells. Obviously, for clinical translation, this is just a small sidestep. We also work on radiolabelable formulations. There we use desferol chelated mice cells. You'll see a clinical example later on. And there we achieve similar things. This image is, I think, 2 days after injection. You still see the heart very clearly. This is a tumor. These are the major blood vessels going up and down to the brain. So there's a lot still in the blood flow. And also here we get like 17, 18, 19% for the radiolabeled formulation that accumulates in tumors. And what is critical, and what was the point of this study, is to show that if we drug load the mice cell, so not only a mice cell that is just a mice cell with a chelator or a fluorescent dye, but as soon as we put thousands of drug molecules in there, does the mice cell still behave as good as an empty mice cell? And to go even further, even if we cryoprotect the mice cell to allow shipping, to promote logistics, and then we reconstitute, and then we inject, do they still behave? Do they still perform? Right? And what you can see in these images here, these are like 3 days after injection now, the tumor accumulation is insignificantly different. So it's always between 50 and 20%, regardless of whether we load, whether we blend, or whether we cryoprotect. One of the issues that I think is massively overlooked in the preclinical field, and that is arguably the most important in the clinical field, is the fact that patients die of metastasis, not of a primary tumor. So we've been studying for many years now, how do nanoparticles, how do antibodies, how do liposomes, mice cells accumulate in different metastases in different organs, right? And we try to understand that from a broad perspective. So we generated some models. This is a model in which we label 41 cancer cells with a near-infrared fluorescent protein, kind of like GFP, but then at the range of 680 nanometers. And we can see that the cancer cells from this primary tumor here, they spread via this metastatic lymph node, right? Which is this huge lymph node. This is a healthy lymph node. This is a healthy lymph node. This is a healthy lymph node. And then they spread to areas of the lung, right? So we see this diffuse cloud of the lung. On the back of these lungs, you would see multiple metastatic nodules. And if we then in the same mouse inject mice cells that are labeled with a fluorescent agent that we can excite at a different wavelength, we can basically see how we reach the tumor, we reach the metastatic lymph node, we reach the lung, and we obviously also here reach liver and spleen, right? Those we cannot avoid. So metastasis targeting does work. We tried to systematically study this to understand how metastases are different between lymph node or lung or bone or soft tissue. We initially looked at more than 100 different metastases. And the first question we asked is, how do metastases compare to the healthy tissue in which they are located? And what you see here is that in lymph nodes, the metastases are always higher than the healthy tissue. In subcutaneous lesions, in the neck, and also in bone areas, metastases always accumulate more than the healthy tissue. In the lung, from ex vivo analysis, I'll show that to you on the next slide, it's the same thing. We just cannot properly construct optical signals in the lung. Because of the air, because of the ribs, because of a lot of fluorescence going through the heart, we have a distortion of the optimal signal. So this is actually a false negative result. If we then again ex vivo, we take out the organs, we scan them for cancer cells, we scan them for mice cells. This is actually the lung where you see the cancer cells in blue, and here you see the mice cells in blue. And now you see that actually in all the locations in the lung where we have cancer cells, we also have mice cells. These are two ovaries, right? We actually show what was the main outcome of this study. Targeting of metastasis is even more heterogeneous than targeting of tumors. We have a big tumor here, we have a small tumor here, small tumor accumulates much more than a big tumor, right? And that's not a consistent pattern. Like in the next situation, it could be the other way around, right? And if compared now to the primary tumor, this is the primary tumor from which all the metastases develop, and compare that to metastasis in this model, which is 41, which is orthotopically implanted, and we just let it metastasize over time, we see that the amount in the tumor is always twice as high as the amount in different metastases, and that the amount of metastasis is essentially always the same. So there's not really any difference if the metastasis is in the liver. Many people think that if something goes to the liver, you get more localization because nanoparticles go there. That's not the case, right? And it's the same for all the other metastases. And the key question here, we looked at many different pathophysiology features. I will not go into details, but we try to understand what feature drives accumulation and what feature makes the difference between the primary tumor and the lymph node to become established. We look at like collagen, we look at macrophages, other immune cells, we look at vasculature, at lymph, and so on. What we found in lymph node metastasis versus primary tumors in this 41 model is the only significant difference is the amount of collagen cross-linking, right? Collagen is in blue here. We initially thought we have more collagen, but it's actually a different structure of collagen. So this is second harmonics. This is not an antibody staining. And with second harmonics and lysyl oxidase levels, which are the collagen cross-linker enzymes, we found out that metastases are stiffer. So the collagen is cross-linked, that makes the lesion stiff, and a stiff lesion compromises vascular function, and it compromises nanoparticle permeability and penetration and distribution into the metastatic lesion. This is the reason why metastases are less easily targetable than primary tumors. When we think about theranostics, again, now go to the clinical situation. This is work done with Iris Miedema and Willemien Menke in Amsterdam. This is a theranostic micelle loaded with docetaxel and labeled with zirconium-89, injected into patients with multiple metastatic lesions. We see that in one patient, this is where the tumor is, right? The tumor is encircled here in computed tomography. This is where the micelles are. Amount of accumulation is somewhere between 3, 4, 5%, but the distribution is not great. So there are big parts of the tumor that do not accumulate micelles. So if you would have enough patients to choose from, this would, in a clinical trial, probably be a patient you want to exclude. Here you have a situation where you have two tumors in the lung of a patient with colorectal cancer. Both of them have more than 10% of the micelles in the tumor, and it's quite nicely homogeneously distributed. This would be a lesion, or this would be a patient that we would definitely want to include. What is important for clinical diagnostics or theranostics is that we understand dosing. Many particles, especially also pegylated liposomes, when you decrease the dose by a factor 100 to make it a diagnostic dose, their circulation kinetics will change because you do not saturate the liver. So you get more rapid clearance from the bloodstream and also lower tumor accumulation. What we found for these micelles, because they are 100% pegylated, each of the 1,000 polymers in the polymeric micelle has a PEG in the hydrophilic shell, that if we down dose to a diagnostic dose, 1% of the treatment dose, the blood kinetics in patients are the same, and the tumor accumulation is the same. So we can actually use them at a companion diagnostic dose. And what many people don't realize, and this is a comment on work of Warren Chan and colleagues published. Bennu Yang was the first author of the paper in Nature Materials a couple of years ago. I was one of the reviewers of that paper, and they were looking at how there is a certain particle dose threshold that determines lung circulation. Normally, when we dose nanoparticles or drugs, we always dose at drug equivalent. So you give 5 milligrams per kilogram doxorubicin equivalent of a nanoparticle. What Bennu Yang and Warren Chan were actually arguing is we need to look at the number of particles we inject. And that in a way makes sense, because a Kupffer cell doesn't care much how many drug molecules are on the inside, but it cares on the absolute number of nanoparticles it takes up until it is saturated. And one of the questions I asked during the revision of the paper was, if you look at your initial meta-analysis, the one I showed to you before, about the 0.7%, what about nanoparticle dosing in that setting, right? And they basically replotted this. And this is another replot from what they plotted in the paper. You see that everything that is above this threshold, this is 10 to the 12 nanoparticles. Below 10 to the 12 particles per mouse, you have low tumor accumulation. Above 10 to the 12 nanoparticles per mouse, you have high tumor accumulation. Right? So the particle number seems to be more important than the milligrams per kilogram of dose that you administer. And something that I looked at when preparing this comment is how does that look in the clinic? When it's really striking to see that if you upscale this number from mouse to humans, 10 to the 12 would be 1.5 times 10 to the 15 nanoparticles in humans, right? Considering the size of the liver and the blood pool and so on. And then out of the clinical formulations that were tested and trialed, everything that is above this threshold, Myoset, Onivite, Doxil, they all made it. They all became products. And those formulations that are dosed below this threshold, they all failed. And here again, the interesting thing is that for the mice cells I reported to you before at this companion diagnostic dose, this study, we're actually below the threshold. And that is probably because this is all dependent on protein corona formation. Liposomes are only 2 to 3% surface pegylated, like half of the 5% peg is on the inside. That means there is still a lot of room for proteins to dock and to promote excretion via liver and spleen. Whereas typically mice cells are 100% pegylated and they do not build this classical protein corona. That means they're not affected by reduced dosing. Right? And I think this is fundamental information for people considering clinical translation and companion diagnostic. Good, so let me skip this. Just say based on this slide that the fundamentals of metastasis targeting are really not well understood, that we should spend much more time coming up with good metastatic models. This paper, we had a figure that was really dedicated towards which models do we have to understand which part of the metastatic cascade. And the other thing that I guess we should all really be aware of, and this is really often misunderstood, is that the location of the tumor does not determine targetability. There are many people that said that if there's a tumor in the liver, because there are so many nanoparticles going to the liver, that also the tumor in the liver can be targeted. I'll show you in a couple of slides that that is really not the case, because the tumor is completely different. The tumor does not have Kupffer cells. The tumor does not have a space of this. Right? The tumor is a mass of cells that is much more cellularly dense than a healthy liver. And then on top of that, if you have a liver metastasis, that's completely different from a liver tumor. So depending on where it comes from, what is the origin, what is the timeline of its growth, it might have a completely different vascular and TME tumor microenvironment makeup. So we should be really critical in terms of understanding the fundamentals of metastasis to understand which of those we can target with nanoparticles, with CAR T cells, with antibody drug conjugates, with whatever your favorite therapy is. Before I move to the tumor priming parts, the second part, 3-4 slides on better alternatives, not better, more pragmatic alternatives to deal with patient selection, with patient stratification. So nanotheranostics, without a doubt, are the most accurate ways to predict, to monitor if a nanoparticle goes to the tumor. We get quantitative feedback on the amount that accumulates in tumors and at the whole body level, also in metastasis. But they're not that easy to do. Right? What is easier actually is if we would do it with a companion diagnostic that does not depend on an individual dose of a long-lived isotope that needs to be shipped, that does not depend on radio labeling and quality control, etc. You just take a nanoparticle as a companion diagnostic, and there are different ways to do this. Right? So you can do different types of low or the classical companion. What I think is the smartest of all of these is if you take another nanoparticle that might have a different size and a different PK profile. It might not circulate as long as a liposome or a micelle, but it still tells us whether tumors in patients can be reached and can be targeted. Right? The numbers might not match perfectly, but it's good enough to say this patient should be included, this patient should be excluded. And there are few, but I think very, very nice examples in the literature of this. This is one of them where a company, this is not from us, this is from a company in the United States, Merrimack. They used iron oxide nanoparticles that are used a lot in the preclinical and clinical literature. Many people think they're approved as MRI contrast agents. They're actually not. There are 12 different iron oxide nanoparticles, all approved for iron deficiency anemia. So they're actually used to reconstitute iron pools in the blood of patients with anemia, and particularly with patients with underlying kidney disease. However, what you can do is you can take those iron oxides, this drug product that is used for anemia, and you actually use it for MRI imaging, right? Because it produces very strong contrast in MRI. And that's what they did in this study. So they looked at many different patients with many different cancer lesions, and they looked at the tumor accumulation of the iron oxide nanoparticles at 24 hours. And then you have a cohort that has good accumulation above the median and a cohort that has low accumulation. It's below the median. And if you then look at the response to a therapeutic liposome, right? This is Onivite, developed by this company Merrimack Pharmaceuticals. It's irinotecan and liposomes. It's used in the clinic for pancreatic cancer therapy. What they could show is that patients that are above the median, that are good tumor accumulation iron oxide nanoparticle tumors, those tumors on average decrease in size, right? So those are responders. Tumors that are below the median, that have poor tumor uptake of iron oxides, also do not really respond to unified liposomes. And again, the heterogeneity is obviously large. Like these tumors, the response are all over the place. But if you now think from a clinical trial point of view, you develop a new company and you want to predict whether your therapy works. If using such a off-the-shelf companion diagnostic repurposing tool, which you could use basically in every patient from tomorrow on, you could remove all of these patients from your trial and only include the ones that show uptake, that show tumors that are accessible, then it's much more likely that your phase 2, phase 3 clinical trials will be successful, right? That's the point of why we do patient stratification. Good, and then an even easier approach may potentially be just to look at biopsies, because we know that from all cancer patients, apart from a few numbers of brain tumors, we always take a biopsy first. So we put a needle into the tumor, we extract tissue, we determine whether it's cancer or not. We look at proliferation, we look at receptor status, mutations, but we can also look at features that determine whether tumors can be delivered to, right? Like what about vessels? What about macrophages? What about cells and so on? And we did this in a very systematic setup and in multiple different animal models, at collagen, at angiogenesis, at lectin, etc. We started off with 3 tumor models in which we used one of our favorite delivery systems. It's an albumin-sized polymer. It's kind of like a long PEG, but then HPMA has the advantage that you can anchor multiple things. PEG only has like 2 ends, so you can maximally functionalize it at 2 sides. HPMA has multiple functionalization opportunities, but here we took the cleanest version. So it's a single polymer with either 1 or 2 size 7 molecules per polymer, and that circulates really long and it targets tumors extremely efficiently. The point here was to show in mice that we have different features in different tumors. So we had one tumor model, A431, with low tumor uptake, one ovarian carcinoma with intermediate, and then one colon carcinoma with very high uptake. Right? So you have 4% of the injected dose for one, 8% for the other, and 10 to 12 for the third one. And in those tumors, we then stained for 23 different features: macrophages, blood vessels, lymph vessels, collagen 1, collagen 3, collagen 4, all of those parameters, and looked for features that correlate with nanomedicine accumulation. And one of the most striking correlations was macrophages. Right, so this is macrophages levels F480 in mice. We use CD68 in human samples. Low in the low accumulation tumor, intermediate in the intermediate, and high in the high. Right, and then we do a correlation, and we see a really nice correlation, like 0.8 R-square value. It's a really nice correlation between macrophages and nanomedicine delivery. And then what we did to do this some sort of in an it's not a non-blinded manner, but let's say in a neutral manner, we basically fed all the data into a machine learning model, gradient-free boosting, and looked at all the 23 features. And what we ended up with is 6 features statistically significantly correlated with delivery, and 5 of those are related to blood vessels, and the 6th one is macrophages. And if you think about that, that makes a lot of sense, because blood vessels are needed to get something to the tumor, and macrophages, in the case of nanoparticles, are needed to keep the nanoparticles there. So what we then ended up doing is we formulated, or we came up with a biomarker score in which we multiplied blood vessels and macrophages. We gave a score of 1 to absent, 2 to low, 3 to medium, and 4 to high. And then we multiplied the score to have a good range for biomarker differentiation. So low blood vessel, low macrophage would be score 4. High blood vessel, high macrophage would be score 16. Then we look from the 10 different tumor models, right? These are PDX models and CDX models, from very high to very low levels of accumulation. We prepared multiple different sections, we stained them, and we gave them to blinded reviewers, including 5 pathologists. So people did not know what they were looking at. They just gave a score of 1 to 3, 4. And then we plotted all those 300 scores in this density map or in this heat map in function of a decreasing order of liposome accumulation. This is area under the curve. This is the highest liposome accumulation. This is the lowest. And you really nicely see this differentiation, where you have the true positives, the ones that are high and high also have a high liposome accumulation. And clinically most important, the ones that are low in blood vessels, low in macrophages, they also have low liposome accumulation. So we can basically predict from the biopsy of the patient whether they can whether the tumors in that patient can be targeted with nanoparticles or not. And I think putting this in a broader context, now we know nowadays that besides EPR and leakiness, there's also transcytosis. There might be blood vessels like nanoparticle transport endothelial cells that are specifically responsible to take in nanoparticles or to transcytose nanoparticles. Maybe these cells have specific markers, and then we wouldn't stain for CD31, which stains all blood vessels, but would stain for CD-NTEC, which is the specific marker for the nanoparticle relevant endothelial cells. And then we can gradually refine this protocol to become even better at predicting what happens. Good, so in the second part of the talk, I'd like to switch to engineering tools to prime tumors. Tumors are often quite hostile. One, they're very heterogeneous. And second, they're very difficult to properly get into because blood flow is not good, because the matrix has a lot of collagen and so on. So one of the key things that I find most important to share with you today is that we overlook tumor perfusion. When we think about drug delivery to tumors, everybody starts with permeability, right? Like blood vessels should be leaky. But what if there is no blood vessel, right? Like there are a number of tumors, I could show you pathological data, probably in the backup slides, where we see hardly any blood vessel in a really big tumor mass. And tumors survive quite well in hypoxic conditions, right? They shift to anaerobic metabolism and so on. Then there are tumors like this example, in which we do see blood vessels in the pathology, but there's no blood flow. So the vessel is there, but probably because there's mechanical forces compressing the blood vessel, there is no blood flow. This paper is actually not about perfusion. It's about a technology we developed, which is called ultrasound bubble tracking or ultrasound localization microscopy. You can basically follow ultrasound contrast agents, which are microbubbles, and you can follow their path and you can determine their velocity, etc. This is a tumor in a patient, a breast cancer patient, approximately 5 × 2 centimeters. It's quite a chunky tumor. And only this small part here that is color-coded is having blood flow. Right? Just think about that. There's 90% of this tumor, very tiny parts here and there. But the vast majority of this tumor does not have blood flow. So even if we would now make a super nanoparticle, how can we guarantee that it will work? Right? And if you think about it even longer, in the clinic, we don't monitor blood flow in tumors, and we don't try to improve it if there is no blood flow. So we just take for granted that blood flow happens, and that will make any type of drug we develop accumulate in tumors. But blood flow doesn't always happen. There are many situations where it's not the case. So we need to image that using engineering technologies, and we need to find ways to improve it if it's not good enough. So we can do this with various different tools. You can use radiotherapy. That was my doctoral studies back in the days. We can use ultrasound and microbubbles. We can use hypothermia. I think with ultrasound and microbubbles, we have a lot of tools now at hand to already make an impact on clinical practice. Microbubbles are used to monitor things like stenosis or infarctions in the heart or in the carotid artery. With contrast agents like Sonovu there are 7 or 8 approved all over the world. They're air-filled or gas-filled vesicles, 1 to 5 micrometers in size, that have a coating of either lipids or polymers or proteins or sugars. So there are different formulations, and what they essentially do is they oscillate in an ultrasonic field. Ultrasound is a pressure wave, right? So it becomes less dense, more dense, and a bubble in such a pressure wave will grow or shrink. And if you think of a bubble in a blood vessel, it will grow and it will shrink. And that means that on the one side, if we have a lot of mechanical forces closing the blood vessel, really pushing from both sides on the blood vessel, that means there is no flow in this direction because the tumor basically pushes the blood vessel to close.
Recording stopped.
Then such a microbubble
Recording in progress
can actually grow and shrink to reopen this blood vessel, to create flow again. The other thing we can do, and I'll show you examples for both, is we can open up the endothelium if there is no leakiness, right? And like I said, perfusion is overlooked. This is an example from a real patient, a study we did with Roel Dekkers in Utrecht in cats. This is not a tumor model. These are old cats that have real tumors that developed over years. We monitored with ultrasound whether there is blood flow in the tumor. You can see that the bottom compartment of the tumor has good blood flow. The top compartment has no blood flow. What we then do is we inject microbubbles. We incinate with ultrasound the top part of the tumor. And then you see how also this top part now starts getting blood flow. So we're basically temporarily, maybe for a couple of minutes, maybe for longer, we're opening this compartment now for delivery of drugs. And if we now have the right drugs in this setting, we might be able to efficiently kill a bunch of cancer cells to reduce the mechanical pressure. And then the blood vessels will stay open for a much longer period of time. This is how this can work in patients. This is a triple negative breast tumor in a patient. 4 or 5 centimeter tumor. This dark mass here is the tumor. This is Doppler ultrasound imaging. So this shows where blood is flowing. And at the starting time point, there is no blood flow in the tumor. Every alternating minute, 2 and 4 and 6 and 8 and so on, for 10 cycles, we apply microbubbles and ultrasound. And then gradually over time, you see how now the tumor really shows good blood flow. And we can actually switch from treatment mode to imaging mode. So we can basically stop once we see we have enough blood flow, and now we infuse drug. And if we would want to, we could even add another dose of microbubbles and another dose of ultrasound later on if we would want to. Good, so this was perfusion. The other thing is permeability and penetration. These things are oversimplified. This is how we normally draw the tumor targeting effect, right? There are big gaps in blood vessels. In real life, it's much more like this. Like, not all blood vessels are leaky, and even if there is leakiness, nanoparticles, they don't swim in a tumor. There is no physical force, there is no convection or diffusion that make those particles go through the tumor. And on top of that, the tumor is very dense, right? So in real life, this is how it looks. This is a blood vessel in a very stromal tumor 3 days after injection. All of these liposomes, they accumulated via the EPR effect or via transcytosis into the tumor, but they don't penetrate. They just sit right outside of the blood vessel. Also here, if we pre-treat tumors with microbubbles and ultrasound or co-treat them, we can actually enhance this penetration. This is a complicated slide, but just look at the two images in the middle. These are whole tumors excised from a mouse. These are ultrasound treated. These are not ultrasound treated. And these are optical images for liposomes injected into a mouse. And this was a case control study because we had 2 tumors in the same mouse. Only one of them in green was treated with ultrasound, the other one was not. And those have more than twice as much accumulation, in some tumors at least, more than twice as much for the ultrasound versus the control case. I think a factor 2 is nice, but not great. The really big impact here is at the level of penetration. So this is without ultrasound treatment. This is similar to the image I showed you before. All these green liposomes, they did go out of the blood vessel, but they just sit right on top of the blood vessel. They don't get into the tumor. So if we apply ultrasound and microbubbles, we see that liposomes move away from the blood vessels much, much deeper. And if we quantify this in 3 dimensions, we go from 90% of the liposomes stuck to the blood vessel wall to almost 60% of the liposomes moving into the tumor, beyond 10 micrometers away from the blood vessel wall. Good, and then in a brain setting, it's obvious that we have this so-called blood-brain barrier that makes it very difficult for materials to move into the brain. What we can do in a brain setting is we can open up the blood-brain barrier with ultrasound and bubbles using helmet devices like this, which are really a highlight of electronic and biomedical engineering, where you have 1024, for instance, ultrasound transducers, symmetric, that can fire in a very controlled manner ultrasound pulses into tiny, tiny spots in the brain. Right? Getting across the skull is actually quite easy. Many people think that's difficult. It's difficult very close to the skull. Deep in the brain is fairly easily manageable. And then you have small hotspots of 1 to 3 millimeters where you can excite microbubbles to overcome drug delivery to the brain. This is just an example that the approach works. Just look at these 2 images here on the right. You see a blood vessel in the brain that has been previously injected. The mouse has been injected with 70 kilodalton dextrane. So again, albumin-sized sugar molecule, no extravasation in the brain if you don't apply ultrasound and bubbles. If we do apply 5 minutes of ultrasound on top of 1 dose of bubbles, you see a lot of the dextrane getting into brain tissue, enabling delivery to brain parenchyma. What is critical here is to do this in a safe way. Ultrasound is very, very potent. You can cause a lot of damage very easily. What we did in this particular study is we optimized the settings of the ultrasound and the dosing and the timing of the microbubbles to conditions in which we do not see any side effects at all, not at the whole body level at the mice, but also with pathology afterwards. We don't see bleedings, we don't see infarctions. We basically don't see anything in terms of side effects, and still antibody-based polymers or antibody-sized polymers and also even 100 nanometer liposomes, they do manage to get out of the blood vessels into brain tissues. 2 more small examples of microbubbles, and then I'm done. So what we can do with microbubbles is we can, on the one side, we can drug load them. If we want to have controlled release only in certain sites of the body, what we can engineer is, well, we work with polymer-based bubbles. The advantage of these bubbles is they have a very thick shell. Polybutyl cyanoacrylate, it's a clinically approved polymer in conditions of wound healing. So you use it as a surgical super glue. It looks kind of like this. It says like star speed glue. So if you have a surgery going on and you have a wound that doesn't stop, what surgeons do is they just drop a couple of drops of PVCA glue, and then you basically glue it in the body, and that's perfectly biocompatible. If you take this super glue and you put it in a stirring solution in which you have a surfactant, can be Triton, can be many other things, we end up with bubbles, right, that basically form at the interface around, in this case, if we stir vigorously, around an air interior. And then we can do many things to the polymers. This is the good thing about polymers. They're very diverse. So we can add antibodies, we can add peptides, we can load drugs inside, etc. And the examples that I want to show with you today is one is this example where we drug load them and we antibody target them. So we basically do two different things. We put a model drug on the inside of the shell and an antibody on the outside. And the concept here is we can then inject those bubbles. They will bind to something on the inside of the blood vessels, like the VEGF-R receptor. And then the blood vessels, even if they're leaky, the microbubbles are so big, they're 1 to 5 micrometers, they would not extravasate. But they create leakiness, right? That's what microbubbles do. That's what I showed you before. And by doing this, we can let them bind. We give ultrasound pulses, the bubbles burst, the blood vessels open. And at the same time, the microbubbles release these agents that they contain. So we basically locally open the vessels for specific drug release in situ. If we look for model compounds, this is just coumarin as fluorescent dye, we get up to 2 million molecules per individual bubble, right? So the space is not big, but it's 100, 150 or so. This is 10. If we do this with step microscopy, it's like 200 nanometer shell thickness. So I guess the best average would be 100, 150. It's quite a big space for drug molecules to go in, right? So it's in the order of millions per individual bubble. And if we give an ultrasound pulse, like 80% of what is inside of the shell will eventually come out. Right? So it's a very efficient local release system. And if you now check how this works in real time, this is a real-time video, you see how these microbubbles in real time bind to the blood vessels in the tumor. Right? This is a replay of the video. It's not like a pulse or something. It would look the same for the pulse because the contrast would also disappear. This is at time 0. This is at time 10 minutes. Right? These microbubbles have a very short half-life, 1 minute or so. But everything that means that at 10 minutes after injection, everything that is in the tumor creating ultrasound signal is a microbubble bound to the blood vessel wall. And if we then don't give an ultrasound pulse, what would happen is you would have such a microbubble bound to the wall of this blood vessel. And if we do give an ultrasound pulse, we actually see that we release the cargo, the rhodomin in this case, from the microbubble, and that is able to extravasate into the tissue surrounding the blood vessel. Another cool thing we can do with polymer-based bubbles is we can play with their shape. This is something that has never been explored before. And what we did here is we took these PVCA microbubbles, the round ones, we put them in a polyvinyl alcohol film, like integrated. Then we heat the temperature above the glass transition temperature of the bubbles. We stretch them in 1 dimension or even in 2 dimensions, we can do this. And then we cool down again. And then we end up with non-spherical rod-shaped or in 2 dimensions, even disc-shaped bubbles, which have the advantage of performing margination in blood flow. Margination means that if you have objects in blood flow in the presence of red blood cells, what will happen is that non-spherical shapes would be pushed towards the wall, right? And that's where we want the bubbles to be present, to be able to open the endothelium. So this is how these microbubbles look. These are the spherical ones. These are the stretch ones, right? They look like some sort of a bacteria. They have an aspect ratio of 2.5. With the pure PVCA polymer, this is the best we can do. With other polymers, we can go beyond this in aspect ratio. If we put them in blood flow phantoms, this was done with Paolo De Cuzzi in Genova, we see how they move away from the center of the blood flow, more towards the extremities, towards where we would have the endothelium in vivo setting. We see that macrophage uptake is delayed. So macrophages are known to have frustrated phagocytosis. That means they try to engulf the microbubble on let's say, the long side, and that is very difficult. So it takes them much longer to actually take up one of these bubbles than one of these bubbles. You can see that here quantified. And because of this frustrated uptake by macrophages, we have a somewhat longer circulation time, from like 2-3 minutes for the spheres bubbles to like 4 or 5 minutes for the rod bubbles. And that helps us to better open the blood-brain barrier. right? So we give either the sphere bubbles or the rod bubbles. We co-administer a dye that does not cross. This is a cross section of the brain, so it's a microscopy slice. This is the normal situation, ultrasound and bubbles. This is where we take the non-spherical bubbles. So it's much more efficient to open up the blood-brain barrier. And then another thing we can do, I mentioned before, we can modify the surface of the bubbles with antibodies, in this case, an anti-transferrin receptor antibody. So this binds to the blood-brain barrier. And then the advantage is we can wait. So we don't have bubbles in blood flow. All the bubbles we have at 10 minutes are bound to the endothelium. That's where we want them. The ones that are non-spherical would bind in higher amounts. And then we apply ultrasound. And now this is what we get under these conditions. This is for the control. Right? So it's really, really potent to localize where you have the bubbles in action to permeate the blood-brain barrier for drug delivery. I'll show you two more examples that are not from us, and then we're done. It's 2 slides. This is how this looks in the clinic, right? This is done in the clinic in patients, not on a day-to-day basis, but in initial clinical trials. This was arguably the very first study ever done in a cancer patient, breast cancer brain metastasis, in which you see a slice through the patient with CT. This is the eye, this is the eye, this is the nose. This patient has a big metastasis centrally located in the brain. And then we inject an HER2 antibody labeled with indium, and we use SPECT to monitor. This is without MRI-guided focus ultrasound. This is with. And here you see how you have a 5-fold more accumulation of the antibody in the metastasis as compared to a non-treated case, right? At 2 days after injection of the antibody. And then last but not least, this was published last, I think, December, maybe November. And I think this is really, really impressive. What people did here, and it's a big consortium, you can see it's a whole big of different people from engineering to clinical medicine teaming up to show that in glioblastoma treatment, adding microbubbles and ultrasound to standard Stupp protocol treatment, you can get enormous enhancements in efficacy. Right? This is really unprecedented. I think this is super spectacular. So what people did here is they took the normal Stupp protocol. That means a patient with a glioblastoma tumor first gets surgical removal at week 0, then 6 weeks of radiation therapy plus temozolomide, which is a drug that is used in glioblastoma. It's not a great drug, but it crosses the blood-brain barrier. That's why people use it. And then here in time in weeks at month 3, month 4, 5, 6, 7, 8, people add ultrasound and bubbles. And then they wait up until 5 years, right? Like this is not even the end of year 1. They wait for really long. And what they found is that the median survival time, if you treat with ultrasound and bubbles only in this period, right? We're treating here, only here, month 3 to month 8. This is time in months now. That the median survival goes from 18 to 19 months to 31 months. So you basically have 1 full year of overall survival added to the patient. That's impressive, right? There's no drug that ever did anything close to this. And on top of that, and I think it's even more impressive, if you look at the 5-year survival, we know for glioblastoma, this is like 4%, 5%. If you add ultrasound and bubbles, you get 35%. So you 7-fold, 8-fold enhance the amount of long-term survivors. By adding ultrasound and bubbles in year 1 of the treatment, 5 years later, you still have a 7-fold survival benefit. And this to me shows that we're massively involving the immune system, right? This is not a drug effect. Drug delivery plays a role for sure, but probably shaking up the tumor from the inside with bubbles and ultrasound will create a situation in which immune cells start recognizing, start acting upon the tumor mass more efficiently. And this can really achieve things that so far have really never been achieved in this particular cancer. All right, so for those of you interested in the subject matter, I'd like to point out 2 papers. I think both of them have, they have unique elements to them in the sense of how they were done. This is what they call a viewpoint. A viewpoint is some sort of a questionnaire. They had 4 different people. Sangeeta Bhatias from Boston is a biomedical engineer. Sean Shen is a radiochemist, a nanotechnologist. Marina Drobovskaya is an immunologist from NIH, and I'm a mix of pharmacology and pharmaceutics. And they ask 5 questions to all 4 people. So you get 20 answers to why do we have so many papers, so few drugs? What about the animal models we have? What about exosomes? What about nanodiagnostics? Like, is there a market or is there a medical need for classical nanoparticle diagnostic agents, etc.? So it's an interesting overview if you want to learn more about where cancer nanomedicine is. And then this is a follow-up with that. This was only Marina and me. I'm not sure why they didn't ask the other 2. This was really on the aspects of translation. Like, where do you see roles or potential for immunoengagement, or how to best balance dosing and PK, etc. So they have 7 different questions, but it's the same type of format. And I think it's fairly insightful to really have different perspectives. We come from very different fields, and the majority of answers, they go in the same direction. So it really shows that at the higher level, or let's say at the longer times of experience, people do agree that certain things need to be done and other things do not need to be done. All right, so with that, I've come to the end. My main points are that tumor targeting definitely works in patients for nanoparticles, for antibodies, for many different formulations, but we need to consider the microenvironment. We might have to prime it for better delivery. And we need to take into account that it's very, very variable. It's a lot of heterogeneity. We need protocols as simple as possible and as pragmatic as possible to find the right patients to treat. We should entail or encompass metastasis when we want to understand how to better treat cancer. We should consider perfusion. It's really overlooked, and it's a really fundamental parameter in all aspects of drug delivery. Priming with ultrasound, but also we worked with other types of pharmacological or physiological priming. They all hold potential in different directions. And what I didn't talk about, there are many people interested in nanoimmunotherapy. This is definitely something that is coming big time. There was very promising data reported 2 weeks ago at the ASCO 2026 on personalized mRNA vaccines, with 5-year survival times increasing 50% in stage 4 metastatic melanoma if you add an mRNA vaccine on top of checkpoint blockade antibodies. So also nanoimmunotherapy is really coming up, and it's really here to stay. With that, Professor Rossi, I'd like to close the talk, and I'll give it back to you. Thank you very much.